What is Creutzfeldt-Jakob disease? Creutzfeldt-Jakob disease is a rare, degenerative fatal brain disorder to be caused by an abnormal isoform of a cellular glycoprotein known as the prion protein. It belongs to a family of human and animal diseases known as the transmissible spongiform encephalopathies or prion diseases. A prion derived from protein and infectious causes CGD in people and animals. Spongiform refers to the characteristic appearance of infected brains which become filled with holes until they resemble sponges when examined under a microscope. CJD is the most common known of human TSE. CJD is caused by a protein known as a prion. Infectious prions are misfolded proteins that can cause normally folded proteins to become misfolded. The disease can be transmitted from an affected person to others, but only through an injection or consuming infected brain or nervous tissue. There is no evidence that CJD is spread through ordinary day-to-day contact with those affected or by airborne droplets, blood, or sexual contact. The disease was first described by German neurologist Hans Gerhard Creutzfeldt in 1920, and shortly afterward by Alphonse Maria Jacob, giving it the name Creutzfeldt-Jakob disease. There are three major categories of CJD. First is the variant Creutzfeldt-Jakob disease. Is caused by eating the meat of cow with prion in the muscle tissue. In cows, this prion cause bovine spongiform encephalopathy, which is commonly called the mad cow disease. Where sheep were fed cow meat, the prion causes the disease scrapie. If a person eats the meat of infected cow or sheep, the prion get absorbed through the intestine into the bloodstream. After that, things is unclear. It's thought that the protein can somehow get through the blood-brain barriers and enter the neurons by the process called adsorptive endocytosis. Adsorptive endocytosis is a process where the plasma membrane of nerve cells fold inwards to bring the substances that could otherwise not be able to cross the plasma membrane by themselves. Next is the sporadic Creutzfeldt disease, without a clear cause, but it's been suggested that a normal brain protein changes abnormally or misfolds and torn into a prion. This is how it looks like. Next is the familial Creutzfeldt-Jakob disease. Happens when there is a mutation in the PRNP gene. The most common mutation happen at the 200 codon and causes the amino acid glutamic acid to be replaced by a lysine, and that's enough to make the entire protein misfold. Let's proceed to the pathophysiology. Spongiform encephalopathy can be broken down. Spongiform means sponge-like. Encephalo refers to the brain. Pathy refers to a disease process. Spongiform encephalopathy is a disease where the brain tissue degenerates and healthy tissue gets replaced by cluster of tiny, liquid-filled cavities called cysts, making the brain looks like a sponge. The cause of a spongiform encephalopathy is the accumulation of misfolded proteins called prions. But first, let's review. Proteins are made up of a long string of amino acids. The exact sequence of this amino acid is called the primary structure. These long chains of amino acid can fold to form a different shape like alpha helix and beta plated sheets. Each protein can contain multiple alpha helices and beta plated sheets. Now, there is a protein called prion protein or PRP, which is included by PRNP gene and is made up mostly in alpha helices, and it's most commonly found in the cell membrane of neurons. Although the function of PRP is unknown, it's thought that might play a role synapses between neurons and the uptake of copper into the cell. When the prion protein misfolded, it changed from mostly having alpha helices to having a lot of beta plated sheets. This abnormal protein is called the prion. When the misfolded protein enters the cells of the nervous system and interacts with the normal prion protein, it accesses the templates, induces the misfolding in a normal prion protein. These prions are also highly resistant to broken down to proteases. As the result of this misfolded prion protein cannot be easily broken down, they also cause normally folded protein misfold and become just like that, and they having affinity from the brain. This misfolded prion protein accumulates within the cells and triggered apoptosis, which is programmed cell death. This is done with the helps of 14-3 proteins, which is the intercellular protein that promotes apoptosis. When large number of cells start to die off, cysts start to form in the brain, and this gives the brain spongy appearance. Additionally, the prions aggregate together on the membrane of the neurons, forming large plaques that are toxic to brain tissue. Now, the most common cause of spongiform encephalopathy is the Creutzfeldt-Jakob disease, or CJD. Epidemiology. CJD affects about 1 individual per million per year worldwide. Approximately 350 cases are diagnosed annually in the United States. Sporadic CJD is the most common form of human prion disease, and the age onset is 61 years old. Death occurs in nearly 70% of patients within 1 year of onset. The signs and symptoms of CJD. The first symptoms is the rapidly progressive dementia, leading to memory loss, personality changes, and hallucination. Next is the myoclonus. The involuntary movement occurs 90% of cases, but may be absent at initial onset. Other symptoms include anxiety, depression, obsessive compulsive symptoms, and psychosis. Also, there is a physical problems such as speech impairment, balance and coordination dysfunction, or ataxia. Magnetic resonance imaging, or the MRI, produce a detailed image of the brain and can show up abnormalities, particularly to the CJD. Next is the electroencephalogram, records brain activity and may pick up abnormal electrical patterns seen in the sporadic CJD. Lumbar puncture, test for a certain protein that indicates you may have CJD. Tonsil biopsy, check for the abnormal prions found in the variant CJD. Genetic test, a simple blood test to find out whether you have a mutation in the gene that produces normal protein. Last is the brain biopsy or autopsy, the only way to confirm a diagnosis of CJD. Currently, there is no treatment for CJD. Supportive care should be given, and genetic counseling should be offered for those with familial form of disease.
In March of 2011, 42-year-old Michelle experienced a sudden onset of stroke-like symptoms, difficulty speaking, confusion, and odd movements. All tests came back negative, and she was sent home, but Michelle was still struggling. In a few days, Michelle was back in the hospital to stay. Stroke and meningitis were ruled out. Kip says primary care doctors and specialists were stumped, and then, without the chance to ever say goodbye, the diagnosis, Creutzfeldt-Jakob disease, CJD. There is no treatment, and it is always fatal. Normal infectious protein called a prion is believed to be the cause of CJD, actually creating holes in and around neurons in the brain. Dr. Axemet says 90% of cases are sporadic, striking spontaneously without a known cause. The other 10% are familial, which was Michelle's case.
And that ends of my presentation. Hope you learned something from the video. Thanks for watching.